I am currently enrolled in a research program put on by the Education and Public Outreach Committee of the Stedman Philippon Research Institute in Vail, Colorado. Our most recent lab in the Center for Regenerative and Personalized Medicine at the Stedman Clinic, utilizing the cell culture lab, we extracted proteins out of mouse cells and used the BCA assay for protein quantification. In this video, I will explain the methods of protein isolation and quantification, the relationship between protein and cell concentrations, and the objective and scientific utility of the BCA assay. Protein isolation is a process of the protein progressing in purity through a step-by-step procedure. The first step involves identification and acquisition of the source, which in this procedure were the mouse cells known as the ATDC5 cells adhered to the well in a mixture of media. The second step of protein isolation involves the extraction of cells from a source. In our lab, we used a triple E enzyme solution to help break down cell-to-cell and cell-to-surface adhesion. This can be thought of as the cells floating in space individually in the solution. Using a centrifuge, we use centrifugal forces to separate the components of the triple E cell suspension. Centrifugation helps us isolate and pellet cells for further research. The remaining triple E solution left after centrifugation is aspirated, leaving behind a cell pellet. The third step is the separation of protein from other components such as lipids and nucleic acids. To do this, we added mammalian protein extraction reagent, or EMPER, to the cells. EMPER is a reagent used to isolate proteins in cells for accurate quantification. The EMPER disrupts cell membranes, which allows the release of proteins into the solution. Then the solution is centrifuged to complete the protein isolation. Following the protein isolation is quantification, the determination of the concentration of proteins in a sample. To do this, we used an assay, which is a test used to analyze concentrations or activity of a substance in a sample. Various assays exist for various fields of study. In our lab, we used a bisinochronic acid assay. The BCA assay uses color change from a reaction when the proteins reduce copper ions in the BCA reagent for a purple-colored complex. The intensity of the color represents a certain protein concentration. Protein quantification is important to scientific research with many objectives and scientific utilities. The main goal is to determine the concentration of proteins in a sample to provide necessary information for later use. Protein quantification is also utilized to compare data and normalize experimental results. In our particular lab, the protein quantification is important when monitoring cell health and viability. Protein disease can be detected to understand the underlying causes. Protein quantification is important when researching drugs to examine the impact of substances on cellular proteins. This enables for the development of drugs and medicines. In conclusion, protein quantification is very important for answering biological questions and researching disease and drug interactions.